So the things that it was understood that this DNA molecule can do then is to hold instructions to somehow make things like brown hair, right? And what causes brown hair, we'll get to this in a little bit, it was understood as actually proteins. So the instructions that this nucleic acid molecule has is instructions to make proteins. That's how it produces traits, right? Proteins cause traits like brown hair. But then they also realized that this DNA had to have somehow the ability to be used to make copies of itself so that it can get passed on to the next generation of cells, and the next generation of offspring, right? Because offspring come from cells. Offspring are made of cells. So that's the story. By 1947, right, it had been about 80, 90 years since this chemical DNA was discovered. So some cell biologists were working on, like what it's made of, even though they didn't know how important it was at the time. It had been figured out that this was a polymer, that it was a polymer of little units called nucleotides. Remember what's in a nucleotide? We have this sugar, this 5-carbon sugar called ribose or deoxyribose, right? We have a phosphate attached to that, and we have one of these things called nitrogenous bases. Remember, those are the things that have more than one nitrogen in a ring. Sometimes there's 2 rings, sometimes there's 1 ring, but these are, it was discovered, the only thing about a nucleotide that's different is this, kind of like the R group of an amino acid, to make an analogy, right? All nucleotides have this and this, and what's different about the nucleotides is this, and there's only 4 of those. Remember, there were like 20 different amino acids, 20 different R groups. There's only 4 of these, and we use those letters to symbolize these chemicals, adenine, thymine, guanine, and cytosine. So that was understood, that basic part of its structure, what the components were. And then this guy added a key finding, French guy, Chargaff. He discovered that when he analyzed DNA from any kind of source, a human, a goat, a pine tree, or whatever, he found an interesting mathematical kind of thing. Here's human DNA that he studied. These numbers are right from his studies. Here's human DNA from his studies, and he found that the percentage of adenine was statistically identical, right? Hard to measure these things, so it's not going to be identical, but statistically identical to thymine. The percent of thymine equaled the percent of adenine. It wasn't always like 30% like here. That's human. It could have been 23% if it was a pine tree or whatever, but it was always the same, adenine and thymine, and so were the percents of guanine and cytosine. So this was figured out, but its significance was not put together until a few more years later. But it was a key, significant, this is going to be on test stuff, this kind of math is going to be on test stuff. It was a key, significant finding leading to the understanding of how DNA works. Okay, so again, 1952, it was determined for sure, oh, it's proteins, it's DNA, not proteins, that's the hereditary stuff. You had these different scientists working on it, we'll tell their story in class, right? But this ended up being a very key picture to figuring out the structure and brought a little more drama to the story of its discovery. These 2 guys and another ended up winning the Nobel Prize for this. We'll tell that story in class, right? But what this picture told them is key things about the structure of this molecule. One, that it was a double helix. It was helix. 2 strands of nucleotides that were wound around each other, connected to each other, and then spiraled, helix, right? That's one thing it told them. It also told them how wide this molecule was. Now, this is not the molecule, it's kind of the molecule. This is a picture taken by a very fancy kind of process that uses X-rays, not electrons, not light, to get an image of a molecule. Now, I'll tell you about this molecule in class a little more, but what you need to know is that it helped them figure out the structure, Mr. Watson and Mr. Crick. And so we'll see that story on DVD. Here's the very vague EOC syllabus things, the basic molecular structure, right, and primary function of the four major categories of biological molecules. That's what that says. The basic molecular structure. Well, how basic do you want me to be? That would be my question here, right? You must be able to identify, recognize each of the monomers. Now, we've already done this, but now we're going to focus on DNA more. But you already had to, remember, be able to identify nucleotides, the monomer of this particular kind of stuff we're talking about now. Okay, so here's just again, here's these bases, thymine, adenine, guanine, and cytosine. Notice again, they consist of rings, sometimes two rings, sometimes one ring. But these rings have two nitrogens in them, and you won't find that anywhere else. So that's a good identifier. Okay, so now it was figured out from Chargaff's stuff and from that picture, it was figured out a very obvious reason why the percentage of adenine was the same as the percentage of thymine in any sample of DNA you come across. Watson and Crick simply figured out, I know why that is. They're always attached to each other, and only to each other. A kind of phenomenon that the word complementary was chosen to use to mean. Complementary means they have atoms in all the right places to form hydrogen bonds with each other. Adenine and thymine have two such places. Guanine and cytosine have three such places. That's just detail. But because of where these places are, guanine won't connect with thymine or adenine, only cytosine. This was a big key to the structure of this molecule, right? And now this aspect of the molecule structure, anti-parallel, will be more apparent when we get to the next picture. So here we have three different models, right? Here we have models where every kind of atom is represented over here. That one doesn't tell us much, to be honest, but you can see the clear spiraling here. You can see the spiraling here, right? You can see the complementary base pairing. That's the term here. Again, A, T, C, and G are called the bases. Nucleotides are not bases, so don't call these nucleotides, and don't call nucleotides bases. The bases are the key part of a nucleotide. So what does this blue ribbon represent here then? This blue ribbon represents the sugar and phosphate part, which make up the sides of a molecule that ends up also commonly being described as a ladder-like shape. So here you see it untwisted with a lot more chemical detail, right? This one shows the most chemical detail. You see A and T and C and G only connected to each other, hydrogen bonded, right? You see phosphate, sugar, phosphate, sugar, phosphate, sugar, right? This, right, is one nucleotide right here. It's connected to another nucleotide by their sugar and phosphates connecting to each other, right? And then these two nucleotides are connected, these two, this one and this one, are connected by hydrogen bonds in the middle, much more weakly, right, by hydrogen bonds in the middle. So now that I've messed this up a lot with our, here's why Watson and Crick referred to their model as the double helix. The two that the word double refers to are two strands this way. This is a strand of nucleotides. This is a strand of nucleotides. The strands are connected to each other in the middle by hydrogen bonds between complementary bases. This double-stranded molecule then helixes up, hence the term double helix. Now that I've made it really messy, if you look at the left side compared to the right side, they're upside down compared to each other. That's quite obvious when you look at the sugar parts especially, right? But even if you look at the nitrogenous bases, the A's, T's, C's, and G's, you see that this G is upside down compared to this G. That's what the term anti-parallel refers to. They're parallel, geometrically parallel, right? Anti means one's upside down compared to the other. This makes a difference in how they're copied, so this does make a difference, which is why we're bringing it up. Okay, so in 1953, Watson and Crick will tell the story, right? Remember, that was just 1 year after it was finally conclusively figured out that DNA was the stuff. Just 1 year after, they published a one-page paper that is maybe the most important paper ever published in biology, and vaulted them right to the top of the all-time biologist list, or very near the top with Darwin. They got to share the top with Darwin. Darwin might be a little ahead of them, right? But they reported that this is here with the evidence that they used to report. We might look at that paper in class. But here are the two very key aspects of this molecule in terms of its function being determined by its structure. Okay? So if this molecule is about information, it's like a recipe book. We've already made that analogy. What's different about a chocolate cake recipe and a meatloaf recipe? It's the sequence of letters in the recipe, right? Because they're all the same 26 alphabet letters. They're just arranged differently in the chocolate cake recipe and the meatloaf recipe. Well, Watson and Crick realized that even though there's only 4 letters in this alphabet, A, T, C, and G, these molecules are exceedingly long. In one of your and my cells, there are about 6 billion of these things we call nitrogenous bases that we use letters to represent. 6 billion in each one of our cells. You can make a lot of recipes with 6 billion letters. And so very simply, if we now have blue eyes here and green eyes here, they said the difference between the blue-eyed recipe you might get from your parents and the green-eyed recipe is the sequence of these 4 bases. That's how it carries information, in the sequence of the bases. How can it be accurately copied so parents can send their blue-eyed information on to little Johnny? Well, they said that that is all about the base pairing. Accurate copying is due to the fact that only A can connect to T and only C to G. We'll get quite more specific about that real soon.